continue with our review of the basic chemistry concepts today. And we'll start by going over the warm-up. This typically what we'll do is we'll kind of start the classes going over the warm-up, because the warm-up's kind of like a review of what we did the class before. So let's do that. Here you can see it's a useful place for you to get a sense of where you are with respect to how the rest of the class is doing. Gives you a little bit of a reality check, right? If you're down here at the 50 or 60%, you can say like, whoa, okay, that's not everybody is there. I need to kind of up my game a little bit if I want to keep up and make it through the class. So again, these warm-up quizzes aren't really worth many points at all. Any one particular warm-up quiz has pretty much zero effect on your overall grade, but they are like little bellwethers, little things that give you and me information about whether you're kind of keeping up or not, and whether you're kind of, where you should be there. So let's take a look in. So which is an example of a negative feedback loop. So again, negative feedback is when there's some change in some variable, the response is going to oppose that change, right? So again, your body temperature is falling, so you do something that's going to bring the body temperature back up, like shivering. This would be a positive feedback loop, but there's, it attracts more and more platelets where there's already platelets. That's going to make things even more intense rather than oppose the change. This would what we call like feed forward, where if you anticipate a change, like with the stomach acid, we're going to see lots of control of hormone secretion with things where things are in these kind of things like this, but this is not feedback, negative feedback. Nonpolar, hopefully, most people got this, do not dissolve in water, particularly after all our lecture and the lab. Like dissolves like. Water is a polar solvent. Nonpolar molecules are not going to dissolve in water. Separate individual water molecules are attracted to each other through hydrogen bonds. Again, most people got that. Which of the following would conduct electricity? Choose all correct. Again, from our lab, we saw that there was ionic electrolytes, ionically bonded things will dissociate into individual ions, which act as charge carriers, which carry electrical current. So the calcium chloride, sodium chloride. 5 people said glucose. That's, if you were one of these 5 people, you got to go back and review and make sure you understand why that's wrong. And you should also go back and probably double check your discussion on your chemistry review lab parts 2 and 3 that we just did, because there's probably something kind of upside down in your discussion or how you're interpreting your results. So,
Professor, can I ask a question about this one?
Sure.
So, I mean, I got the 2 answers correct, but I had also put distilled water. Was it just wrong because it doesn't conduct electricity well, really? Because it still conduct electricity, even if it's
barely,
just distilled?
barely. Like we're going to talk about pH, which has to do with, so water in and of itself, there's a little bit of the H2Os that break apart into hydroxyls and H pluses, but it's tiny. It's like 10 to the -7th. It's like it's like 10 millionths of a proportion. It's tiny little bit of charge that's actually available. Like when you like have the toaster fall into the bathtub and electrocute you or something, that's not just distilled water. There's all
Right,
sorts
right, right.
of other minerals dissolved in there that are going to help make the water more electrically conductive.
Yeah, no, that makes sense. I just was like, it was one of those ones where I was like, well, I mean, kind of, sort of.
I did the same thing on that question, so don't feel bad.
Yeah. So, and again, so I would say conduct electricity well. And in fact, we saw like when I put the electrodes just in water or water with sugar, there was no buzz. So that's also giving you kind of direct empirical evidence that just water is not in and of itself conducting electricity well, where it completed the circuit, so the buzzer went off. So you could kind of think of that as well, that evidence. So when ionically bonded compounds such as sodium chloride are dissolved in water, here, 3/4 of you got dissociated individual ions, but a number of people, like 1/4 of the people, did not get that. This one, we're going to be talking about more today right now when we talk about osmolarity. It's really important that you get this idea that ionically bonded compounds dissociate and break apart into multiple subparts. So like sodium chloride, when it goes into the water, becomes individual sodium ions and independent individual chlorine ions, which then are going to actually exert their own osmotic pressure. So we're going to see that in just a little bit. Any questions about the warm-up?
So is this going to be a weekly practice where we have warm-ups to do before our Tuesday start?
There'll be one before Thursday as well. There'll be one just before every class.
Okay. And then when we're in person, will it continue to be on Canvas?
It will still be on Canvas because
Okay.
I used to do them online and have people swap them and grade each other's, but then I realized this actually is pretty nice. And then in lab or in class, when we're in person, I can just project this up on the screen and we can go over it, in person still. But it'll be something on Canvas to do before you head off to school. Or I guess you could do it on your phone. If you get to school early, you can actually do it as long as you have internet access. But it's a way to make sure that you're keeping up. It's always going to be the material that we went over the class before, so it's just kind of a little encouragement to not let things slide, and also to give me a sense that people are keeping up as well. Okie dokie. The other thing, it's worth kind of going over now is the other quiz, that chemistry review quiz. So let's talk about that as well, since we're here. Chemistry check-in. Again, look at the quiz statistics. There are a fair number of people that were getting less than half of these. If you are down here, that doesn't mean that you're not going to do well in the class, but it means that you really need to review these concepts and get up to speed on them if you are going to do well. So this really is an important thing to pay attention to. If you're up here and you kind of got the stuff and you're just brushing up and getting back up to speed, that's great. But you're not going to pass this class if you don't have a good understanding of the basic chemistry. So I will say that. So if you are down in this 50% and lower region, you need to find what resources are available to get comfortable with these concepts. Again, there's office hours, there are YouTube things. We're going to have tutoring. Usually tutoring has reached out to me by now, and they haven't. So I'm going to have to reach out to them. But there's going to be like tutoring. There's so many different resources, but unless you kind of proactively do that and reach out, this class will not go well. So please don't ignore the chemistry or think like, I'll look it out of the corner of my eye and hope it goes away, because it's just gonna come back and bite you. So let's just go over these questions. Again, I'll do this kind of quickly. And again, this is stuff, when people first took this, we hadn't already reviewed it in class. So it could be people didn't know it when they took this quiz, but now that makes sense since we've done the lab and we've covered things. So in a glucose solution, which type of chemical bond is responsible between the water molecules and the glucose? Again, we've talked about hydrogen bonds, this kind of electrostatic attractions between local charge imbalances. What kind of bond is making between the individual atoms to make up a single glucose? Now, those are covalent bonds. Right, and we've talked about that. And we said in particular with glucose, it's a polar covalent bond. There are local charge imbalances, which makes it a polar molecule, which is why it dissolves in water. Which type of bond is responsible for the attraction of between Na and Cl? It's ionic bonds. Again, you should be able to recognize compounds that are ionically bonded. Things like NaCl, calcium chloride, potassium chloride, basically things that have something from the first or second column of the periodic table, like the alkali metals, like sodium, potassium, and things, the halogens, like the chlorine from the seventh column of the periodic table. Things where things have very few things in their valence shell, things have an almost finished valence shell, they're much more likely to participate in this thing where either totally give up an electron or totally grab onto one electron, versus the sharing that happens in covalent bonds. Which type of bond makes up the attraction between the individual atoms in a single water molecule? So most people got this wrong when they first did this. Right now, you should all know it's polar covalent that connects up the two hydrogens and the oxygen that make up a water. So make sure that that makes sense. That's why water is a polar solvent, because each individual water molecule is a polar covalently bonded molecule. Change the number of electrons. Obviously, we get an ion. Again, when we started the class, only like 60% of the people had a good sense of an ion. Ions are critical in this class. Ions are the way that electrical currents are being carried in the body. We're going to see ions till the last gasp of this class. You have to make sure you really understand what an ion is. Ions are going to be critical players in so many things over the semester. What would happen if you change the number of protons an atom had?
It would change the element.
It'd be a different element. Exactly. Which one would best conduct electricity? We just did this, sodium chloride, because it dissociates into individual ions. This is another thing. I know people take chemistry and come out of chemistry and still don't know what the heck a mole is. Mole is important. We're going to be talking about moles a bunch today. It's going to be one of the main ways we use to specify concentrations, like molarity, osmolarity. So you need to understand what a mole is. In fact, less than half the people here got that a mole of one element has the same number of atoms as a mole of another element. Definitely not mass. Like a mole of hydrogen floats, a mole of lead sinks to the bottom of the sea, right? If you have Avogadro's number, 6 times 10 to the 23rd atoms of helium in a balloon, it's going to go float up into the air. If you have that many atoms of lead, you're not going to even be able to lift it off the ground. So yeah, make sure you realize it is not mass. It's the number of atoms or molecules if you're talking about a compound. Whoop. pH, we're going to talk about pH in more detail today. You know, when pH goes down, the acidity is going to be going up. Lower pH, like a pH of 2 is way more acid than a pH 7, which is neutral. pH 14 is like really alkaline, the opposite of acidic. You know, alkalinity is the opposite direction of acidity. And hydrogen ion concentration is basically what acidity is going to be a measure of. The more you have hydrogen ions floating around in your solution, the more acidic it is. And what we're going to talk about the actual relationship. It's a negative log relationship. pH is just minus the log of the hydrogen ion concentration. So we'll see that in more detail. I'll talk about it in lecture, and then it's going to be something we're going to continue to go deeper into as we do part 1 of the chemistry review lab today. But pH is another critical thing in the class. pH is important, so you need to understand what pH is, and pH can be confusing because the numbers go up and down in non-intuitive ways until you get a sense of it. Covalently bonded molecules. Again, as we started the class, only 1/3 of the people got this one right. This is important. Covalently bonded molecules will sometimes dissolve in water. It depends on what. What is going to determine whether that covalently bonded molecule will dissolve in water or not?
Whether
Polarity.
it's polar or not.
Whether it's polar.
Polarity.
If it's a polar covalent molecule like glucose, dissolve. If it's a non-polar covalent bond that you find in cholesterol, doesn't dissolve. So it really depends on whether it's a polar covalent or non-polar covalent bond. Here we just talked about this. Change the number of protons, you get a different type of element. And then non-polar do not dissolve in water. So that's why fats and steroid hormones and things like that are going to need special carriers and other methods to actually get carried around in the bloodstream, because you know, the blood is basically, plasma is basically water. So if you're going to move non-polar molecules around, which are going to be important, the fats and again, steroid hormones, things like that, you're going to have to come up with more elaborate methods to get them to get into the blood, because on their own, they are not going to want to just dissolve in water. Sugar, no problem. You just add sugar into water and it goes into solution, right? We saw that in lab. Are there any questions about any of these?
Professor, are these quizzes available for us to look over again after we take them?
I'm assuming.
I haven't checked. I was just wondering.
Yeah, I'm looking at mine right now,
okay.
so you should
Okay,
be able
cool.
to.
Thank you, Rebecca.
This one was immediately, you could see your answers immediately after you took it, but the warm-up quiz that we took yesterday, it hides it until I think everybody's is graded, but then you can go back and look at it.
Yeah, the warm-up quizzes, I let the answers show like after we do the lecture, because otherwise you could just, people talk to each other. So this way, but I think I have it set for 5 o'clock on the class day. The answers show up for those warm-up quizzes. So, all right. So guess we're gonna continue on. Lola. I should check. Did Mylena ever show up? Mylena?
Melina? Yes, I'm here.
Oh, you are here now. Okay. Okay. And Marcos. Marcos? Okay. All righty. So let's continue camera review. Right. And again, I am spending like almost like 2 weeks of this class doing this chem review, for 2 reasons. One is I am assuming that if you've taken chemistry, that doesn't mean that you totally have all these concepts super solid just as you're starting the semester. Or sometimes you have to see things a couple of times to either kind of refresh them or sometimes just understand them at a deeper level. And I also want to just make it clear how important these concepts are for everything else that we're going to do in the semester. These are the concepts that everything else builds on. So please don't just think, oh my God, I can't wait till we get past this chemistry review so we can get into the physiology, because you'll just be lost. When we talk about how a neurotransmitter binds on a receptor, it's all chemistry. When we talk about how the neurons create these potential electric voltages that change to signal things, it's all chemistry. When we talk about how cells get energy to do all of the things they got to do, it's all chemistry. So there's a lot of chemistry and physics in this class, basic chemistry and physics, not like really subtle nuanced stuff, but you got to have the basics down. All right. So let's talk about chemical reactions. And I often just say Rexons. When I was taking my classes, that's what my teachers would write, is RXN. And I just keep doing it. It's just easier to write. It's quicker. So if you ever see me write that, it just means a reaction. So chemical reactions, this is where you have molecules, rearranging bonds into different configurations. Let's talk about like the basic types. There's a few main classes of chemical reactions that we should just kind of lay out as we start. What are the basic kind of types of chemical reactions that you tend to see?
Combustion.
Solutions?
Solution is not solution is.
Oh, sorry.
So combustion is a specific type of what kind of reaction? Combustion is kind of part of. And so actually we are going to talk about that as well. So let me write down like decomposition. Also, or catabolism. This is where you're breaking something apart. You have something and it turns into different parts made up of, so like when you're burning sugar, you're kind of breaking it apart. Combustion of sugar, you're breaking it apart. But this is decomposition, or catabolism, is breaking things apart. There's also, synthesis, which is also called anabolism. That's where you take separate things and they can turn into a single thing. You're building things together.
So like single replacement.
So there's like exchange or displacement.
So Professor, I know that in chemistry, like, when we take chemistry, we classify the reactions as like a single replacement and a double replacement.
Yeah.
Would those things be considered an exchange? just
Yeah,
across the board? Okay.
Yeah. So this kind of thing, these are going to be, this will be important. For an example of one of these kind of exchange or displacement reactions that we're going to see a bunch of is phosphorylating things, like phosphate group, usually just write as a P. Adding a phosphate group to something is really common. Like it is how you often control enzyme activity. If you have some enzyme, you can phosphorylate it and now it's active, and you take the phosphate group off and it's not active anymore. So we want to have a way where we can add phosphates to things easily. There is a molecule all over the place in the cell that has a phosphate group ready to donate, which is, ATP. So we're going to often see something like, oh, here's something, let's say some enzyme, and here's ATP, which is actually just A with a P and a P and a P, right? ATP triphosphate. And what's going to happen is one of these phosphates is going to move over to here. So we'll have this enzyme that's phosphorylated, and left over will be ADP, the adenosine with only 2 phosphates left. That would be an example of an exchange or displacement that we'll see a lot, phosphorylating something by moving the phosphate over from an ATP onto like an enzyme or something to control its activity. Does that make sense? I should mention most of the reactions in the body don't go just on their own. They need things that will lower the activation energy to make the reaction actually go. What do we call the special things that lower the activation energy of reactions and can speed up the rate?
A catalyst?
Catalysts. And in the body, that we have special organic catalysts. What's an organic catalyst?
Enzyme?
It's an enzyme. So most of these things are going to happen with some enzyme that's going to facilitate the reaction actually going. We're going to talk more about enzymes in a little bit. Enzymes are not used. That's why I kind of have it kind of entering in and entering out. Like when we're done with the reaction, the enzyme, this is another enzyme I shouldn't, like enzymes that phosphorylate things, we're going to see a bunch of them. They're called kinases. We're going to see kinases start out the whole breakdown of sugar. There's going to be these hexokinases that phosphorylate glucose. In muscle metabolism, to regenerate ATP, we're going to have creatine kinases. In signal transduction with all these second messenger signal systems, we're going to have all these protein kinases that are part of the signaling within cells. We're going to see a lot of kinases, different kinases in different situations as part of different processes, but their job is fundamentally always the same, to drive a reaction that phosphorylates something. So you should recognize this term kinase. It's an enzyme that's involved in phosphorylation. Phosphorylation, like sticking a phosphate group on something. So we're gonna,
Sorry, one quick clarifier. You started to say that an enzyme is not used, and when a reaction is done,
it's not used up. It remains unchanged
okay.
by the end of the process. We're going to talk more about enzymes in a few moments, actually.
And is that specifically an organic enzyme or catalyst? Oh, so
Yeah.
sorry. Yeah.
Everything, all the catalysts we're going to talk about in this class are organic molecules and hence going to be enzymes. In chemical engineering, if you're coming up with these processes for making fertilizer or making different molecules in an industrial setting where you're having these big reaction chambers at super high temperatures, then you're going to use different kinds of catalysts. But all of the stuff that we're going to see in our class are going to be considered enzymes. All right, let's go back to where we were though. What's another kind of reaction?
A redox.
Yeah, redox. Oxidation reduction. These are going to be really common, particularly important for the energy producing reactions. Like when we look at, turning glucose into ATP, it's going to be all about oxidation reduction. What does it mean? What does it mean to oxidize or reduce?
To lose oxygen molecules?
Nope.
Oxidation is loss of electrons.
Yeah, oxidation is loss of electrons. Reduction is gaining electrons. Again, it's a slightly non-intuitive name. It's reduced in charge. If you gain electrons, then your overall charge is going down because electrons are negatively charged. So, oxygen is often involved in these because oxygen has this real, I was talking about the potential energy of electrons in a, maybe it's easier to kind of, I talked about like, I can have like this stapler, or here, this is kind of bigger. Here's like the Choco Cat tape dispenser. I can lift it up high with respect to gravity. The higher I lift it, the more potential energy it has with respect to gravity. And when I drop it, it can fall down to a lower state of energy, and it can do work as it falls. And it can fall lower, and at some level, it's going to fall. It's going to like hit the floor, and there's nothing even below that it can drop to. Oxygen is kind of like the floor for electrons. It's like lowest energy state. Like as you have these redox reactions where one molecule is holding electrons in a high energy configuration and handing it down to another molecule in a lower energy configuration. So the electrons like quote unquote fall, but as they fall, they can release energy. Ultimately, oxygen is going to be kind of the final electron acceptor that is going to be able to accept electrons. And once you're on oxygen, there's not going to be something lower. But it's also why oxygen is really dangerous and corrosive. Oxygen is there pulling electrons. It's like, oxidative damage is one of the major things that kind of damages your cells and things in your body, because oxygen wants to grab these electrons and rips things apart and damages things. We're going to see ways that your body has to try to protect itself from oxidative damage. That's why taking antioxidants is a good idea. The more you learn about oxidative damage, the more you like, oh, I want to make sure I have lots of antioxidants in my diet, because I don't want my My molecules getting ripped apart. All right, let's continue here. So we saw those different kinds of chemical reactions. When we use the word metabolism, this is just the sum total of all the chemical reactions in the body. So that word metabolism just means all of these reactions. The reactions that are making things, the reactions that are breaking things down, the reactions that are interconverting things. So that's all this word really means. Other things we should mention, this idea of chemical equilibrium. I kind of was drawing those little sample equations with these one-way arrows, but most reactions don't go like that. I can have A, B goes to C plus D or something. But most reactions can go one direction or the other direction, depending on the conditions. Right? If you have some reaction and you start adding lots more of this C and D, the reaction is going to start moving that direction. Or if you take these away, the reaction will go back this. We have lots. So most chemical reactions are not one way. They can go either direction. They can go this way or they can go this way. And depending on the conditions, they might move one direction or the other direction and find some new equilibrium, some new balance point where you have some certain amount of the reactants in the products. So this is going to be important. Like when we talk about how buffers work for our lab today, buffers work because of chemical equilibriums. With dissociation of weak acids and bases, depending on the conditions. They'll either more will dissociate or more will reassociate depending on the conditions. So most of the reactions we're going to be talking about aren't necessarily one-way trips. They can go one way or the other way. Some of them are effectively one way, right? If you have D is some gas that bubbles off and becomes lost to the system, then we're obviously not going to be moving the reaction backwards again because the product just floated out into the room. So some things are effectively one way, especially if the products disappear. But for the most part, we're going to see chemical reactions that can move both directions, and which direction they go depends on different conditions, like specifically like concentrations of reactants and products can affect where this balance point is. So chemical equilibrium. We should also talk about reaction rates. So let's say I have some reactions, some reactants, the product. So let's just say it's a one-way thing for just our simplified things right now. What is going to affect how fast, what the reaction rate is that this reaction goes?
Wouldn't that be a catalyst?
So one thing can be catalysts. In fact, let's do catalysts last because it's going to be the most we have to kind of discuss and expand.
Temperature.
So let's start with temperature. Right, temperature we said is just that native energy that the little molecules have. So if you have higher temperature, then these things are going faster and harder and more likely to whack into each other, or they're vibrating or rip apart from each other. So whatever is happening is going to happen faster and more if you have a higher temperature, because all these little things are whacking into each other harder and have more of a chance of having something happen. So higher the temperature, the higher the reaction rate. It's going to be directly proportional to temperature. What other things will affect reaction rate?
What about like elevation?
Say it again.
Elevation, just elevation. I mean, I know it does with boiling water, but I don't know.
Concentration.
Yeah, concentration. Like how concentrated the reactants or the products are. If you have more higher concentration, it's more likely that these things are going to whack into each other and have a reaction. If they are not as concentrated, it's less likely it's going to happen. We're going to see shifting equilibriums by either if we add in reactants or take them away, or add in products or take them away, that's going to cause the equilibrium to shift in one direction or the other. So concentration will affect how fast this goes and even what direction it goes. And then enzymes, which again are organic catalysts. What are the defining qualities of a catalyst?
It speeds up the reaction.
So it speeds up the reaction. If you want to be more formal, it kind of lowers the activation energy. But basically a reaction that normally would not go on its own very fast or even barely at all with an enzyme can go very zippy. What else? There's one other defining quality to a
It
catalyst.
doesn't get used up.
Yeah, it is not used up. When the reaction is over, the catalyst is unchanged, just ready to go again. So, and again, we're going to be talking about enzymes. All the enzymes we're going to talk about are going to be proteins. There are some enzymes that are not proteins. Like Tom Check was like 20 years ago, got a Nobel Prize for showing that RNA molecules can act as enzymes. So not all enzymes are proteins, but all of the main players that we care about are proteins. And that actually is going to be important too when we talk about proteins in more detail later. You'll want to remember that enzymes are made out of protein. And basically the thing that the enzyme works on, we call the substrate. So for instance, let's say this is some enzyme that is going to break apart sucrose into two simpler sugars like glucose and fructose. Substrate, this is just what the enzyme acts on. And then we have the enzyme itself, which I'll call E. And typically what happens, these are going to then go to become some enzyme substrate complex. This is temporary. And then that is going to go, and we're going to end up with some kind of products after the substrate has been dealt with. But then the enzyme is now still just ready to go again. It's unchanged. It's not consumed. It's just there. I'll talk a little bit more about that now. The basic idea, enzymes they're big. They're big, huge molecules. Again, we're going to talk about in more detail. The shape of an enzyme is super important. So it has what we call an active site that is going to bind this active site is going to be able to bind the substrate. So let's say this is some enzyme that breaks apart sucrose. That active site is going to have just the right shape. So when my substrate, in this case, in my little example, like a sucrose molecule, just fits in there. And when I say bind, let me be a little, I'm trying to like, it's a temporary, kind of a temporary attraction. So we're going to be talking about receptors binding neurotransmitters or binding hormones. A lot of times binding has to do with hydrogen bonds. So there's like electrostatic attraction, so it holds in there, but it's not so tight that it just stays there forever. When we say binding in this context, it goes in, it kind of clicks in, but then it can get knocked out again. Think about like a magnet. Actually, yeah, this is perfect. In fact, I don't know if people have like Apple watches. Like you have like your little Apple Watch and it fits on this little charger, which is like a little magnet, and it holds on there and it sets in there and it's there, but it's easy to yank it off. That's a lot what binding is like. It has the right shape. It fits in. There's actually these attractions where it kind of gets stuck on there, but then you yank it off. It's not meant to be a permanent attachment. It's meant to be a temporary attachment where something happens, charging your iPhone, and actually my iPhone has a magnetic charger now as well, for that matter. So the binding in this context, either binding, substrate binding an enzyme, or later on we're going to talk about ligands binding receptors and stuff. It's going to be this kind of temporary attachment that's usually dependent both upon the shape of things as well as the kind of chemical properties, what kind of bond, like hydrogen bonds and things that the things like to make.
Professor, so are you saying that with hydrogen bonds, it is more temporary? It doesn't bind, it's not a strong bind if it's a hydrogen bond?
Yeah, no, I mean, think about, we talked about last week, water molecules are hydrogen bonds. You can take water and you can splash them apart really easy.
Okay.
They're constantly being made and breaking apart and made and breaking apart.
And that applies with the substrate and the enzyme and the
Yeah, exactly.
Okay.
So the shape of this active site is super important. If the shape of the active site isn't just perfect to match the shape of the substrate, then it's not going to fit and it can't do its job. So that is a big thing that we are going to see a bunch of. Anything that messes with the shape of the enzyme is going to affect its ability to do its job. Hold on, trying to figure out how to stop this thing. Nabbit. I apologize for all this. Some of the beeping is you guys, but some of the beeping is me. All right.
So like temperature or like pH could be an example of something that changes that active site.
Yeah, so when we talk about proteins in more detail, when we talk about things that denature proteins, denaturing a protein means messes its shape up. Anything that can denature a protein can inactivate an enzyme. So it's part of the reason why your temperature homeostasis is so important. Your pH homeostasis is so important. If the pH or the temperature are not in the right zone, then it messes with the shape of this thing, enzyme, this, and all of a sudden your chemical reactions aren't going to be functioning properly. So for an enzyme to work, this active site has to have the right shape in order to bind the substrate. Typically, there's something like this, like you have some substrate and you have some enzyme. And remember I said they're kind of separate. And then you have this the enzyme substrate complex where the substrate can go and settles in. Heck. The heck. It binds. But then usually there's some kind of tweaking. As it gets in here, it's stressing out the bonds if this is going to be breaking it apart. So this thing breaks apart. So then all of a sudden this thing floats off as two separate things, but the enzyme is still unchanged and just ready to go again. So, but this binding is the critical part, and the binding, the enzyme binding the substrate will only happen if the active site has the right shape. So again, when we talk about denaturing of proteins and hence enzymes later, that's going to be why denaturing an enzyme stops it from doing its job. If it can't bind its substrate, then it's not going to catalyze the reaction. So any questions about enzymes? Enzymes are going to be another just constant companion for the rest of the semester. Some of them will just talk about that as an enzyme. Some of them will give names. You know, again, I already talked about kinases. We'll meet a variety of different kinases that are going to be involved in phosphorylating things in different situations. There's going to be carbonic anhydrase, which we're going to actually meet pretty soon. There's all sorts of enzymes that are going to be important. And in fact, let's talk about the idea of a metabolic pathway now. So we've kind of got all the pieces to kind of put this into context to how all of these chemical reactions and enzymes fit together in the big picture of all the reactions in your total metabolism. So let's talk about the idea of a metabolic pathway. So from what it sounds like from its name, metabolic has to do with chemical reactions. Pathway, it's like a number of steps you have to take to go from where you are to where you're going. I'll give you an example. So let's talk about cellular respiration. Cellular respiration is something we are going to be looking at in quite a bit of detail. What is the basic chemical reaction that describes cellular respiration?
The ATP.
Well, that's like one of the products. But what's the the overall chemical reaction.
Is it the use of glucose?
It starts with glucose.
The glucose and oxygen to make carbon dioxide and
Yeah, so
ATP.
the 6O2 becomes 6 carbon dioxide, 6 water
Respiration.
plus energy. Right, so this is the chemical reaction that describes the breakdown of glucose. Right, this is just glucose. To ultimately, we're gonna store this energy as ATP when we're doing it in the mitochondria. It's also just this, the description is the combustion of glucose. If you're just light glucose on fire, this is the reaction that you'd break it apart and get carbon dioxide, water. The energy would be light and heat rather than ATP. If you want to make the energy captured in ATP, you got to get more complicated than just lighting the glucose on fire. So when we actually do this in the cells, it's not so simple. Even though this is the reaction that describes the breakdown of glucose to make ATP, we're gonna spend almost an entire lecture describing all the different steps to make this happen. It is going to be so many different steps. In the end, you're gonna start with this and you're gonna end with this, but it's not gonna be a simple equation like this. You're gonna start with glucose. That's going to be our starting point. The first step is going to be phosphorylating the glucose. This is definitely not ATP, right? This is a glucose that's been phosphorylated. Any reaction that's happening along a metabolic pathway is going to have an associated enzyme that is going to help it run. In this case, we're going to have an enzyme that we call a hexokinase. Hexo means working with hexose, 6 carbon sugars. So it phosphorylated a 6 carbon sugar, we have glucose. There's going to be the next step. And then the next step, by the time we're finally done, because I'll say this is like, A, B, C. By the time we get to Z, this is going to be like our ATP. There's going to be all sorts of steps involved. This is what we mean by a metabolic pathway. And each metabolic pathway is going to have its own enzyme. It's going to be enzyme B. This is going to be enzyme C. Right, so we're going to start with glucose for sure. We're going to end with ATP, but it's not just a simple single reaction at all. This metabolic pathway is many different steps, from step A to step B, and from B to C, from C to D, And in this, I'm saying Z, it's not necessarily 26 steps, but that's just to give you a sense, it's many steps. We're going to look at the steps in more detail later. So this is what we mean by a metabolic pathway. And this is almost all of the reactions that we're going to be looking at happen more like this rather than as a simple one-step kind of thing. Any of the compounds that's made that's not the final compound is called an intermediate compound. right? This phosphorylated glucose, it's not we don't care about it in and of itself. It's just one of the steps along the pathway to eventually get where we want to go. So these intermediate compounds are made along the pathway, and the intermediate compounds then turn into yet something else along the metabolic pathway. So does this basic idea of what an metabolic pathway is make sense? You have your initial step, your kind of reactant, your product, but then all these intermediate compounds that are made along the way as you go from one step to the next, all these different enzymes. This is just an enzyme here that take you from one intermediate compound to the next intermediate. So does that make sense? So part of why this is useful, like let's say I have like A to B to C, dot, dot, dot. Let's say Z, we said is like ATP. This is the glucose. There's all these different steps. Each step, there's some enzyme. Each of these are intermediates. What's nice about these metabolic pathways is they give you a lot of flexibility to do different things. Like if a cell has enough ATP already and you're taking in more sugar, what happens? What does your body do with this extra energy coming in if you don't need it because you already got enough ATP?
Stores it.
It stores it. How do you store energy?
You break it down into something else.
Like, what's one of the classic storage molecules?
Fat.
Fat. So at some point, you know, let's say it went from A, B, C, D, E, F, G, H, I, J. Let's say here we're at K and it continues on J, H, I, J, K, L, or whatever. There might be a thing that goes, instead of going to K, we go to K prime, into L prime, and dot, 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 and we end up with fat at the end. So we start the first steps, the first like 10 steps of the breakdown are all the same. So we have the same pathway, but then there is an off ramp. And instead of continuing on the steps to end up with ATP, we take an alternative path now, which I'm calling like K prime, L prime, which ends up making fat and storing that energy that was originally in the glucose is now stored as fat. So we can have an off ramp. Also, as you know, you can make energy from other things that aren't just glucose. You can make energy from starch. You can make energy from fat. You can make energy from breaking down proteins. So, you know, maybe I have protein here. You've eaten protein, and there's going to be these steps like A prime, B prime, C prime, D prime. But ultimately, the last steps are going to be the same. So we can have this on-ramp. The first steps of breaking down a protein to make energy are going to be unique, but then eventually you merge onto this, the final parts of this pathway, which are common to whether, it doesn't matter whether you're breaking down protein or glucose for energy, the final steps are always going to be the same. So does this make sense? Right, and we're going to talk about different ways your body has of controlling enzymes, turning enzymes on or off. So you can actually, like at this point, this branch point here, how does the cell know whether it should be going down this path or this path? It's going to depend on what enzyme is turned on, whether the enzyme takes you to the next step, to the K intermediate or the K prime intermediate, right? So you can, by controlling enzymes, turning enzymes on or off, you can direct this. Also by the concentration of products. We talked about concentration of products affecting the rate of reactions. If particular intermediates are starting to build up, that's going to probably keep this from just continuing in that direction, and it might go another direction. So there's different things that can affect which direction you go. Someone had a question?
Yeah, sorry, I had a question, or yeah. So I know, so like people, when you were talking about people who don't have the right enzymes to process certain proteins, so that like an example of that would be like lactose intolerance, right?
Correct.
So when somebody and I don't really know how this works, which is why I'm asking, but people like take medicine to help them, I guess, process dairy. I know it's not like 100% effective, I don't think, but like,
Well,
are they basically taking like fake enzymes?
they're not fake, they're real enzymes.
Oh, real enzymes? Okay. They're
Yeah,
not like,
and I mean, yeah, I mean, an enzyme isn't, there's no such, I don't think there's such thing as a fake enzyme.
well, I was just guessing, I guess, because it was not being created in their body naturally, that maybe we had chemically engineered them, but I guess they're just other enzymes that have
Right. and
organic enzymes that.
Right, and you'd have to have something that is not, that can survive the acid of the stomach and stuff. So I'm not sure exactly what, but so if you mean by fake, like not exactly the endogenous enzyme, maybe it's not exactly the same enzyme version that would be in your body, but yeah, so maybe the word fake is throwing me off a little bit.
But it's just, they just take enzymes and that's kind of
Yeah,
the
that's one way you
solution.
could, yeah, that's one way you can do it. If you add the enzymes to break down the stuff that then you're cool.
All
Like
right.
if you're lactose intolerant though, and you don't break down the milk sugar and it makes it all the way down into your large intestine where all of these bacteria live that totally can break it down, you're gonna feel kind of crappy.
All right. Yeah. All right. That's pretty neat.
So compounding on Alexandra's comment, so why would you take, so I understand what she's saying about a synthesized enzyme and not something that's organic and produced by the body. So when you take an enzyme that's over the counter coming from a bottle, why do some work better than others?
I don't know enough about how that works. In general, just being in the stomach is a really, really harsh place. Like most enzymes do not survive very well in the stomach because of the low pH. So that's probably part of the reason. But I don't know. I've never really looked into that. So I can't really answer that well. Maybe that could be something you do your research project on.
Here you go.
All right, so I am going to just give you a sense here. Like, this is a sense of what like these metabolic pathways look like. Metabolic pathways, in this case, we have here glucose and one step, a next step, a next step. Notice there's different places you can go. The one we're going to look at, we're going to start here. We're going to go down here to pyruvate, to acetyl-CoA, into the Krebs cycle. So we're going to look at this in a lot of detail in our class. This is what happens in the cytosol and the mitochondria for the kind of most vanilla version of cellular respiration. You can see there's lots of on-ramps and off-ramps in this process here, but way more overwhelming than this is, if you really look more in detail, this is like a picture of the metabolic pathways of the body.
That's awesome.
Each of these little things is a intermediate with some. So if you were in a biochemistry class you'd spend the whole semester just learning a lot of these different pathways and what they are and how they interconnect. That's what biochemistry is. Luckily, that's not our class. We're going to focus on a few select metabolic pathways, specifically like cellular respiration, which is useful to understand in more in depth. A few other ones that are going to be useful to think about. But just to put into context this idea of metabolic pathways, your body is this crazy mosaic of these multi-step chemical reactions that all kind of interreact with each other and have on-ramps and off-ramps to each other, depending on the body's needs. So, when we get to school next week, I have some really nice posters I can bring into class so you can take a look at these and get a better sense of them rather than looking at them as kind of little illegible things on a screen. So questions about idea of metabolic pathway. All right, so we are going to be talking about metabolic pathways. Make sure you understand what it means. What is an intermediate? That there are specific enzymes that will take you from one intermediate to the next intermediate. All right. We need to talk about specifying.
Real quick about the, is the end product always ATP of a metabolic pathway? Or just in these examples?
Even in these examples, it's not. Remember, I said if you take an off ramp, you might make fats.
Oh, okay. Got it.
Right. And a lot of these metabolic pathways, they are describing all of your chemical reactions. So some of them are about making who knows what, or breaking things down, or.
Got it. Okay.
Yeah. Yeah, no, in fact, very few of them are making ATP. All right, so we talked about solutions, and we talked about there were the true solution. We talked about colloids, suspensions. How are these? So these are all some kind of solute, which is then dispersed within some solvent. And in almost everything we're doing in our class, the solvent is water. And the solute is whatever this little thing is that you are dissolving in the water. Maybe I'll make it black so it's easier to see. What's the difference between a true solution versus a colloid versus a suspension?
The size of the particle?
The colloid is cloudy.
Colloid is cloudy. It's we call
like in our
lights.
lab that we did last week. Would the starch solution be considered a colloid?
It is.
Okay, that's what I put in my notebook.
True solutions they do not scatter light. They're kind of glass-like when you look through them. How are true solutions and colloids similar to each other?
Well, those two will stay in solution the whole time.
Yeah,
They'll stay separated.
They stay in solution.
Or I'm sorry, they'll stay combined the whole time. Whereas a suspension will eventually settle to the bottom. It's like the example you used last week was, say, like mud.
Yeah.
You can shake it up and it'll distribute, but then it'll eventually settle.
Or even blood, mud and blood, right?
Blood.
The blood
Yeah.
cells will eventually settle on the bottom even. So suspensions settle. So we're going to be pretty much everything we're doing in this class is in solution, dissolved in water. So we need to be able to specify concentrations. Well, I'm realizing it's 9:30, so let's take our little break. I'm going to just finish up specifying concentrations of solutions, and then we'll do the lab. We'll kind of review what pH is so we can do the pH buffer part of the lab, and then we will take another break, and then we'll start the jumping lab as well, for that matter. So we've got, yeah, I think we're going to wait until Thursday to really dig into the different biomolecules. We're almost done with kind of the more general things about chemistry, but then we're going to have to talk really specifically about, proteins versus carbohydrates, versus lipids, et cetera. So we're going to be spending a lot of Thursday going more into detail about what exactly is a protein, what's an amino acid, what's the amino group or the side chain or the carboxyl or whatever, what's a lipid versus a carb, what's a monosaccharide versus a disaccharide versus a polysaccharide. All right, but let's take a little break, then we'll come back into this, and then we will continue with our chemistry review lab.